Okay, Chemistry 3111. This is the second to last chapter in this class, which deals with amino acids, peptides, and proteins. So we're going to figure out what a protein is and a peptide. And of course, in order to figure out what those are, we have to start with amino acids. And you can probably guess what kind of functional groups are found in amino acids just by looking at the name. In order to master the content in this chapter, it's probably a good idea to review the following concepts. So make sure that you are confident in your Kahn-Ingold Prelog rules, assigning RNS configurations to chiral centers. Make sure you're familiar with the structure and reactivity of carboxylic acids. That's found in chapter 20. Nucleophilic acyl substitution, also found in chapter 20. And of course, the properties of amines, because we're dealing with amino acids. Anyhow, so again, you want to make sure that you're confident in all of these topics before you start embarking on studying the content in chapter 25. Well, let's talk about what proteins are for a second. And if you think about the word polymer, if you walk up to somebody on the street, they definitely know what would be able to tell you what a polymer is, and they'll probably say, well, a polymer is something like a plastic. Okay, maybe a garbage bag is made out of polymer, or maybe a CD case or a Tupperware container or something is going to be made out of a plastic polymer, and that's definitely true. However, those are synthetic polymers. Proteins are naturally occurring polymers, and the monomer that makes up the polymer, a protein, is an amino acid. So if you've ever wondered what proteins are, they're simply amino acids linked together. So a bunch of amino acids. And if you're wondering what an amino acid is, well, I have two amino acids shown here, and the author has represented them as puzzle pieces, and you can kind of link them all together. What do you notice about the amino acid? Well, I notice that it contains an amino group. It's shown here, and we'll look at a bigger structure on the next slide. But we have an amino group, and we have a carboxylic acid, don't we? We have a carboxylic acid here, so a carboxyl group. And that's where the name amino acid comes from. So an amino from an amine, and an acid meaning a carboxylic acid. So those are the two functional groups we find in an amino acid. If we look a little closer at the structure of an amino acid, it says here alpha amino acids. So alpha, the Greek letter alpha, amino acids. The amino and carboxyl groups are separated by one carbon, and we call that carbon the alpha carbon. So you can see here in the puzzle piece that the amino group and the carboxyl group are connected by one carbon. And this is what we call the alpha carbon. Here's a bond line structure of an amino acid here, and you can see again that the amino and the carboxyl groups are separated by one carbon, and we call that the alpha carbon. And so that is why we call naturally occurring amino acids, we call them alpha amino acids. Now, something else you probably noticed about that alpha carbon is that it's attached to four different substituents. If you focus on the bond line structure here on the right, you notice that this carbon is attached to an R group, an amino group, a carboxyl group, and there's also a hydrogen, which isn't shown. And so that is a chiral center. And it says here all alpha amino acids, except for glycine, and we'll look at what the structure of glycine must be, are chiral. The alpha carbon is a chiral center. So chirality is going to play a big role in this chapter because alpha amino acids, except for one, are all chiral. All right. Well, before we get into amino acids even deeper, what you're going to see is that we're going to look at the 20 naturally occurring alpha amino acids. And they're all going to have this generic structure that I have shown here on the right. Okay, and they're going to have all of this in common. They're all going to have an amino group. They're all going to have a carboxyl group. They're all going to be attached by an alpha carbon, the carbon highlighted in green, and they're all going to have an R group. If you look at the DNA of all the people in our class, and you were to sequence it, you would see that we have a lot more in common than we do, different from one another. So for lack of a better way of explaining it, the way that I explain the 20 naturally occurring amino acids is like this. They all have, basically this generic structure here. But what makes us all different? What makes all the students in this class different, right? It's we could say our personality, right? We all have a different personality. And I'd say that the R group is what gives the amino acid sort of its personality. And by personality, I'm talking about its behavior, its reactivity, its physical properties. Anyhow, what happens if we join those amino acids together to make a protein like we had shown on the previous slide? Well, if we only join a few amino acids together, we get what's called a peptide. So again, we join some amino acids together through amide bonds. And it says here that relatively short amino acid chain, meaning having less than 40 or 50, we call that a peptide. The prefixes di, tri, and tetra are used to describe a peptide of a specific length. So say, for example, you have 2 amino acids linked together, you would call that a dipeptide. If you had 3, you'd call it a tripeptide. If you have 4, you'd call it a tetrapeptide, so on and so forth. When we have a polypeptide, that refers to a chain of fewer than 40 to 50 amino acids. Peptide bonds are the amide linkages that connect the amino acids. If we go right back to the first slide, we go all the way back here, you probably noticed that where all the puzzle pieces connect, you see where they connect right here? You can see there's an amide bond here. We know that an amide is a carbonyl with a nitrogen attached to it. Now these nitrogens in this puzzle pieces here, they all have a hydrogen attached, but we have an amide bond. In this chapter, instead of always calling it an amide bond, oftentimes I'll just call it a peptide bond. So you can see that where I have the blue circle, we have an amide bond or a peptide bond. We have another one here, we have another one here, and we have another one here. And so every amino acid that is bonded to another amino acid is held together by a peptide bond or an amide bond. So the peptide bonds, again, are the amide linkages that connect amino acids, specifically this bond and this bond. You see that I have sort of 3 pieces of an amino acid, or 3 pieces of amino acids rather. I have 1, 2, 3 here colored in green. Well, you can see that they're connected by those bonds that I have highlighted in yellow. Those are amide bonds, aren't they? Absolutely. You can see that I have my carbonyl and my nitrogen attached to it. But in protein chemistry or amino acid chemistry, we simply call those peptide bonds. Larger chains are often called proteins, though the term protein implies biological function. So the word protein, I think, means something like of first importance or something like that. But just because you have a long chain of amino acids linked together doesn't necessarily mean it's going to have a biological function. And you can actually destroy the biological function of a protein. You do that if you burn your skin on a cookie sheet, for example. Okay. Anyhow, and we'll talk about that a little bit at the very end of the class. But again, a protein does imply that there's some kind of biological function. So we link all these amino acids together, and then magically we get some kind of biological function by putting a whole bunch of amino acids together. And we'll look at examples of proteins and enzymes throughout this chapter. Well, what about the structure and properties of amino acids? Again, I told you that there's 20 alpha amino acids that occur naturally, and they differ in the identity of their R groups or their side chains. And so the way that they're going to be named is going to be based off of their R groups or their side chains. If you go back to the carbohydrates chapter, we saw that naturally occurring sugars were D sugars, like, for example, D-glucose or D-fructose. But natural amino acids are the opposite. They're L amino acids. If you look at the Fischer projection of L-glyceraldehyde, you see that the hydroxyl at the chiral carbon, or the chiral carbon that's furthest away from the most oxidized end, but there's only one chiral carbon anyhow. But this chiral carbon, you see that the hydroxyl is on the left-hand side in L-glyceraldehyde. If you look at the Fischer projection of an amino acid, an alpha amino acid, what you're going to see is that the amino portion is going to be on the left-hand side. So that's an easy way to remember. As long as you know that a D sugar had the hydroxyl on the right-hand side, and that naturally occurring amino acids are L amino acids. If you draw their Fischer projections, there's only one chiral center in most alpha amino acids. Some of them have more than one chiral center. We'll look at examples of that. But the alpha carbon being a chiral center, except for in the case of glycine, the amino group is going to be pointing to the left-hand side. So here are examples of two different amino acids. Again, you don't have to have the names memorized, but as you practice the problems throughout the chapter, you will familiarize yourself with the structures of a few amino acids. Remember that generic structure that I showed you? The generic structure looks something like this. We had our carboxyl group. We have our alpha carbon, which had a hydrogen attached. We have our NH2 group, and then we have what? We have our R group. If the R group is a methyl, when R is equal to methyl, that amino acid has a name and it's alanine. And of course, the stereochemistry here makes it an L amino acid, so this is L-alanine. If the R group is a CH2, so a methylene with a hydroxy attached to it, then it's called serine. You can see how the names alanine and serine have no connection to a methyl group or a CH2OH. So it's purely memorization or familiarization of yourself with the R groups in order to know the names of the 20 naturally occurring amino acids. Again, you don't have to have them memorized, but you will familiarize yourself with them as you progress through the chapter. Well, let's look at those 20 amino acids and look at all of these different R groups. And here we have the naturally occurring amino acids drawn as bond line structures. They're not represented as Fisher projections. And you can see that all the chirality centers that have the amino group, you can see that they're all drawn as a wedge so that it's coming out of the page like that. Anyhow, something else you notice here at the bottom is that it says each one of these amino acids that's on this page, and there's only 9 of them here, but each one of the 20 naturally occurring amino acids has a name. For example, alanine, we looked at it on the last slide. Not only does it have a name, but it's got a 3-letter abbreviation and it's got a 1-letter abbreviation, right? 20 naturally occurring amino acids and 26 letters in the alphabet. So we have enough letters. Some of them are abundantly clear, like alanine is A, valine is V, glycine is G. However, some of them are not as abundantly clear. Say, for example, if you look on the far right here in the middle, you see phenylalanine is the letter F and tryptophan uses the letter W. Well, what do these amino acids on this slide have in common? These are all amino acids with non-polar side chains. So aliphatic side chains, if you will. If we have a hydrogen as our side chain, that's not a chiral amino acid, is it? Right? Because we know that there's already another hydrogen attached here. So the R group is in essence, or is an actual hydrogen atom. And so this is an achiral amino acid, and you heard me allude to that earlier. I said there is an amino acid that isn't chiral, and so the name of that amino acid is glycine. It's the simplest possible amino acid. I'll go over the rest a little bit quicker. If I have a methyl group, CH3, it's alanine. If I have an isopropyl, okay, then I have valine. I remember that because it kind of looks like the letter V, like that. So valine is an isopropyl. I have valine repeated here. If you go to the next one, this is an isobutyl. When you have an isobutyl chain, that's leucine. When you have a sec-butyl, so this is a sec-butyl chain, here it gets confusing, it's called isoleucine. You also notice that there's a second chiral center here, right? This is one of those amino acids that has 2 chiral centers. Then the next one is a very interesting amino acid because the nitrogen is not a primary amine, it's a secondary amine. So the R group is actually tied back into the nitrogen like that, and that gives proline some very unique properties. The next one is phenylalanine. If you remember that alanine was simply a methyl, if you add a phenyl group to it, well, you've got phenylalanine. The next one is tryptophan. This big ring system here where I have a 6-membered ring and I have a 5-membered ring with a nitrogen in it, this is an aromatic system called indole, okay? Now you might look at this indole and say, well, that's going to be polar because it's got a nitrogen in it, you know. But the thing is that the rest of the molecule is just, or the rest of the piece of the molecule in tryptophan is just so big that the nonpolar carbon-hydrogen bonds overcome that nitrogen-hydrogen bond. And so the side chain is considered to be nonpolar. So tryptophan. Something else that's interesting about phenylalanine and tryptophan is that their side chains are planar. And that has some interesting properties. when you look at the biochemistry of enzymes. Here are some amino acids with polar side chains. Asparagine has an amide on the end. It's got the letter A in it. It's only got one methylene in it. The next one is glutamine. It also has an amide at the end. The letter G comes after the letter A. It's got two CH2s. That's kind of how I remember them, sort of alphabetical order. The next one is a serine. When we have a CH2OH, that's a serine, right? Polar side chains. The next one is threonine, which has another chiral center in it. Tyrosine, I always think of the word tire swing. Okay, you've got a phenyl, right, and then you've got a hydroxyl hanging from it. You can imagine yourself, I don't know. Anyhow, then you have cysteine. So that's the way I pronounce this, is cysteine, because cysteine is something else. Anyhow, cysteine has a thiol in it. So what's the commonality between all these? Well, when your R group either has a hydroxyl or an amide, so we'll put an amide here, or a thiol, okay? If you have either a hydroxyl, an amide, or a thiol, that's considered a polar side chain. Next are the acidic and basic side chains. The acidic ones are pretty obvious because they both have carboxylic acids hanging out at the end. You have aspartic acid and glutamic acid. If you go back to the last slide, you noticed asparagine and glutamine, right? If you replace these NH2s with OHs to make carboxyl groups, asparagine and glutamine become aspartic acid and glutamic acid. So maybe that helps trying to memorize those. The first two basic side chains, arginine and histidine, are maybe not as abundantly clear as the carboxylic acids. And then the last one just has a primary amine dangling off the end, and that's lysine, okay? And lysine has 4 methylenes, right? 1, 2, 3, 4. It's handy to memorize that because when you want to draw the structure of lysine, you want to draw the bond line structure. You don't want to write out all of those methylenes explicitly. All right. I don't ask you to memorize the structures of these or their 3-letter abbreviations or their 1-letter abbreviations. However, I will be using the 3-letter abbreviations during the lecture. So if you need to go back and refer to this slide, no problem. If you need to just, write this out on a sheet of paper for your own personal edification, that's no problem too, because it gets really painful if you're trying to draw or write down the structure of a peptide. Say it's a decapeptide. Well, then you've got 10 amino acids that you've got to write down. You don't want to write down the names of each one of them. You just want to put down an abbreviation. Okay. If you're, kind of used to working with amino acids, which I don't do a whole lot, teaching organic chemistry, I don't teach biochemistry. I use the 3-letter abbreviations. When you get to biochem, I'm sure they'll just skip over those and go right to the 1-letter abbreviations because it's so much faster. All right, structure and properties of amino acids. Our bodies can actually make half of the naturally occurring amino acids. Okay, we can actually just synthesize them, our own cells. But the other 10 amino acids, they have to come from our diet. If you're a meat eater, so if you are not, if you're the kind of person who eats meat, fish, milk, eggs, so on and so forth, you're going to get all the amino acids that you need. And we call that the protein that comes from these kind of foods. We call that complete protein because it has every amino acid that you need. Now, if you're a vegetarian, on the other hand, you have to be a little more careful because it says here, no single plant source contains all of the essential amino acids. So vegetarians and vegans need to have a combination of grains and beans and nuts. For example, I think it was the Aztecs who ate, or maybe they invented succotash, or it was invented a long time ago. And succotash is a mixture of rice and corn. And when you mix rice and corn together, you're sure to get all of the all essential amino acids. And the reason why is because rice is missing 2 amino acids and corn is missing amino acids. Anyhow, the 10 amino acids that your body can make, these are called the non-essential amino acids. Anyhow, our textbook has a little bit more information about this, but this is about the depth that you need to know about essential and non-essential amino acids. Well, an amino acid contains an amine and a carboxyl group. You know, it's not that uncommon to have molecules besides the 20 naturally occurring amino acids that contain amino groups and carboxyl groups. For example, gamma aminobutyric acid, or GABA, is a neurotransmitter found in the brain. It's not an alpha amino acid, but it is an amino acid, isn't it? And thyroxine is a hormone that's found in the thyroid gland. You can see that it also has a carboxyl and an amino group. And if you have studied the citric acid cycle in biology, you might have even seen another intermediate in there that is an amino acid that's not a naturally occurring amino acid. Anyhow, well, let's talk about the physical properties of amino acids a little bit more, namely their acid-base properties, right? They got to be interesting, because the amino acid has what? It's got a portion that's acidic, and it has a portion that's basic. I mean, if we draw out the bond line structure, it looks something like this, okay? So this is an amino acid, and this is how they were drawn. If we go back, go back all the way here, we didn't have them drawn with any charges or anything. They just drew the amino group and the hydroxyl group, and we'll get into the charges in a second here, but it says under acidic conditions, or low pH, both the amino group and the carboxyl groups are protonated. Well, why would that be? I can tell you why. It's because the amino group is basic, isn't it? We spent a whole week talking about amine chemistry. So this is basic and this is acidic. This in a strong acid, what's going to happen? Well, the acid isn't going to get protonated again, but the base is going to pick up a proton, isn't it? Right? If we treat that with H+, okay? And they're saying here, low pH. Right? So then we're going to have a positively charged amino acid. And if we look at this positively charged amino acid, okay, now we've got two acidic protons, the one highlighted in red and the one highlighted in blue. And each one of those protons, or each group highlighted above, has its own unique pKa value. If you increase the basicity in which the cationic amino acid is in, or you increase the basicity of the environment that cationic amino acid is in, what you see is that as you start to increase the pH, that the first proton that's going to get removed is the one from the carboxylic acid. So that tells you that it's got a lower pKa. It's a stronger acid. The second one that's going to be removed is the one that's going to come from the ammonium portion, or the protonated amine, which tells you that it has a higher pKa. Well, what are the pKa's of this carboxylic acid and the protonated amine? What do they look like? Well, we've already looked at the pKa's of carboxylic acids earlier on in this class, and they were, around the neighborhood of 2, and that's what we see if we examine the pKa's of the protons on the carboxyls of these amino acids. You can see they're all kind of in the same neighborhood, aren't they? They're all around 2, 2-ish, a little bit above and a little bit below, but they're all in the neighborhood of around 2. So we'll put here the pKa1 is around 2, not exactly, but give or take. And then you can see that the pKa of the protonated amine, the ammonium portion, is what? It's around 9 to 10, right? They all kind of fall in that range. So we could put here around 9 to 10. Maybe I'll color code it. There we go. So somewhere in that neighborhood from 9 to 10. Again, note that the carboxylic acid is more acidic than the ammonium. Now, what else do we see here? We see that we have pKa's associated with the side chains. Arginine, for example, its side chain has a pKa of 12.48, and aspartic acid has a pKa, or its side chain rather, has a pKa of 3.65. Well, we're going to look at the consequences of having a third pKa in your amino acid. Notice that the acidic and basic side chains have their own pKa. Again, for example, we have lysine, right? Tyrosine, okay? When we have a group, an R group that has a proton, it's going to have its own, when we have a proton that's acidic enough, it's going to have its own pKa associated with it as well. All right, well, let's move on from there. How do we use these pKa values? What's the significance? It says here the carboxyl group of alanine, right? The carboxylic acid portion of the amino acid alanine has a pKa value of 2.34. So if we draw the structure of alanine out, it looks like this. We've got our amino group here. We've got our carboxyl group here. And then alanine is just when we have a methyl group. So this proton has a pKa value of 2.34. When the pH is equal to 2.34, the amount of protonated carboxyl and the amount of deprotonated, or its conjugate base, of alanine is going to be equal. So at a pH of 2.34, we're going to have an equal concentration of the carboxylate and the carboxyl. Well, what does that tell us? That tells us that at physiological pH, what's going to predominate? The carboxylate is going to predominate. Why? Because physiological pH is in the neighborhood of around, I don't know, if you ask students, they're always going to come up with a different answer. 7.35 to 7.45, roughly, something like that. So if we compare the pH at which we have an equal amount of carboxyl and carboxylate, and we increase that pH, means it's going to be more basic. That means the equilibrium is going to lie to the side of the carboxylate, right? We're going to deprotonate a greater amount of the carboxyl than we will have protonated. So at a pH higher than 2.34, more alanine molecules will be deprotonated, and vice versa. At a pH lower than 2.34, meaning more acidic, we're going to have more of the carboxyl than we will of the carboxylate. What's the consequence of all this? At physiological pH, the structure of an amino acid is going to be zwitterionic. What is a zwitterion? It's a compound that has both a positive and a negative charge in it. Physiological pH is higher than the carboxyl pKa, which was what? Around 2. And it's lower than the pKa of the ammonium, or the protonated amine, which is around 9 to 10. So again, physiological pH fits in between the two. And so again, at physiological pH, the structures of amino acids are going to be zwitterionic. Now, at physiological pH, amino acids are amphoteric. That means that they can function as both bases or acids, depending on the situation. If you look at these two structures, they're identical. Okay, these are both just a zwitterion or a zwitterionic amino acid, alpha amino acid. When we take that zwitterion and we treat it with a strong base, like a hydroxide base, like sodium hydroxide or lithium hydroxide per se, well, what's going to happen then is that the zwitterion is going to behave as an acid, right? And it's going to donate a proton. Okay, well, the converse is true if we were to treat that zwitterion with an acid, right? If we treat it with a strong acid like hydronium, it's going to protonate it. All right, so these are the kind of things that you want to think about when you're looking at the structures of amino acids. Well, let's define something here called the isoelectric point. Sometimes, for some reason, Mr. Dion, I know what an isoelectric point is. I've studied them for a long time. But sometimes I teach electron configuration so much, I guess, that sometimes I just can't help but say isoelectronic point. Anyhow, it's the isoelectric point, or the PI, is the pH at which the zwitterion concentration is highest. Now, what pH would that be? Well, for an amino acid that doesn't have an acidic or a basic side group, you're just going to take the average of the two pKa values. Again, all of those amino acids that didn't have a third pKa associated with it, all you do, add the two pKa values, divide them in half, and there you go. You should be able to do this without a calculator. I always have somebody who says, do I have a calculator? You should. If you're in university, if you entered a university, you should be able to add two numbers. For example, if I said, we have $2.34 and $9.69. How many dollars do we have? Well, you should be able to add that up. Now, what if I split it between two people? You should be able to do that as well. Okay, so addition and division are things that you should be able to do. Well, what about the other amino acids? The ones that have a third pKa associated with them. Acidic amino acids, the average pKa value is going to be that of the side chain and the carboxyl group. And for basic amino acids, the average pKa value is going to be the value of, it's going to be, for basic amino acids, the average pKa value is going to be that of the side chain plus the protonated amine group. So for example, if we're talking about a basic amino acid, we'll look at lysine, everybody's favorite basic amino acid. You see the pKa associated with the R group is 10.53, which is in the neighborhood of 9 to 10. So it's more associated with the basic residue or the basic the pK of the protonated amine rather than the carboxyl. So we just add those two together and we divide them in two. So we take the average, and this would be our isoelectronic point for a basic amino acid like lysine. If you have an acidic amino acid like glutamic acid, you have two acidic residues or portions in the molecule. You're going to take the average of those two pKa's, and that will be your isoelectric point. See, I almost did it. Okay, so calculating isoelectric point, not very difficult. Now, is there any, consequence of isoelectric point? Is there any, information that I could garner from that, or is there anything useful I could do with an isoelectric point? Yeah, absolutely there is. If you've ever heard of electrophoresis, I'm sure that many of you have tried gel electrophoresis in a biology lab, for example. Well, we can actually separate amino acids by exploiting their isoelectric values. It says here separating amines, but it should say separating amino acids. Okay, so if we look at the isoelectric point of an amino acid, if the pH of the solution in which your amino acid is in, if the pH is lower than the isoelectric point, right, or if the isoelectric point is greater than the pH, whatever, okay, but I like to think about it in terms of pH. If the pH is lower than the isoelectric point, that means it's in an acidic solution, right? It's more acidic. That means that the amino acid is going to be protonated, right? It's going to be cationic. It's going to have a positive charge. Okay, if the pH, on the other hand, is greater than the isoelectric point, that means it's in a more basic environment than the isoelectric point. So you're going to have an anionic amino acid, right? It's going to be more deprotonated. Well, then what's the consequence? Let's say I take these three amino acids, lysine, alanine, and glutamic acid. You have all of their isoelectric points. You see that lysine is a basic amino acid, glutamic acid is an acidic amino acid, and alanine is somewhere in the middle. Well, if you put them in a solution that has a pH of 6, what the heck's going to happen? If you put that solution in between charged plates, where we have a cathode, it's got a negative charge, and an anode that has a positive charge. Well, at that pH, right, think about it. A pH of 6 is lower than the isoelectric point of lysine. So if the pH is lower, that means it's going to be more acidic. So it's going to exist in its protonated form. That means it's going to be positive, right? It's going to be cationic. So it's going to be attracted towards the negative end, right? It's going to move towards the cathode in our electrophoresis experiment. Alanine isn't really going to move because it's going to be right at the isoelectric point, so its charge is going to be completely balanced out. It's basically going to be neutral. It's not going to move. Whereas glutamic acid, on the other hand, it's in a solution that has a pH that's higher or more basic, and therefore it's going to exist in its deprotonated or anionic form, and it's going to migrate towards the anode, the positive end, like that. Now, this is not a way to preparatively separate amino acids, and if you're like, what does preparatively mean? Preparatively means separate enough that you would be able to carry out a synthesis with it or something. It's usually, and I'm not an expert in electrophoresis, but it's usually used to identify amino acids in a mixture. Okay, we use an electrophoresis experiment to either identify them or to quantify how many different are in a mixture. Right, we can only separate little tiny bits of amino acids this way. Again, if two amino acids have similar isoelectric values, well, how would they separate then? Then it's just going to be based off of their relative size. The larger alpha amino acid is simply going to move more slowly. Again, electrophoresis is simply an analytical scale technique. Analytical means a small amount. Okay, it cannot be used to separate huge amounts of amino acids. So if you want to separate grams of amino acids from, then you're going to need another technique. Amino acids are generally speaking colorless, but we can detect their presence by treating them with what's called a ninhydrin color indicator. And there's a really interesting piece written in our textbook about the ninhydrin color indicator and how it's used to detect fingerprints at, say, a crime scene, for example. When we perspire, our sweat is mostly water. Around 99% of our sweat is water, but there is small amounts of amino acids in proteins that comes out in our sweat. And so when a, what's the word I'm looking for? I don't know. Oh, shoot. When a forensic scientist goes to a crime scene, they'll spray this ninhydrin, around where they think there might be some fingerprints. And what happens is it will react with the amino acid in sodium hydroxide to give this purple colored product right here. So this product has a purple color, and then you end up with the byproducts, which are water, carbon dioxide, and the R group of the amino acid. Anyhow, so a way to detect amino acids. Another thing I should point out about amino acids is that since they're zwitterions, that doesn't show a zwitterion. Where's a zwitterion? Here's a zwitterion. Since they're zwitterions, it's like it's as if they have a similarity with an ionic compound, right? They have a positive and a negative charge. And so they do share some physical properties with ionic compounds, namely that amino acids are going to be solids at room temperature. They're usually beautiful, white crystalline solids. And also they're going to have high melting points. So similarities with ionic compounds. With all that in mind, I think it's time. It's 8:40 in the morning. Let's try a practice problem. Be a perfectly legitimate question for the quiz. Question is, although most naturally occurring amino acids are made up only of L-amino acids, or most naturally occurring proteins rather, are made up only of L-amino acids. Proteins isolated from bacteria can sometimes contain D-amino acids. Draw the Fisher projection for D-alanine and D-valine. In each case, assign the stereochemical configuration of the chiral center. Well, if we go back to glyceraldehyde, okay, if you go back to glyceraldehyde, CH2OH, and this is kind of where my thought process goes. Okay, we had the hydroxyl on the right-hand side, right? This was D-glyceraldehyde. Okay, well, that means that our D amino acids are going to have their amino group on the right-hand side. Well, with that in mind, let's try drawing D-alanine. So if we have D-alanine, like this, we're going to make our Fisher projection. I'll help you out with this. The carboxyl is going to be at the top. This, H2H. We're going to have our hydrogen over here. We're going to have our amine over here. And then our R group is going to be on the bottom. We already looked at a Fisher projection, or a couple Fisher projections earlier on, but probably need to have your memory jogged already, which I wouldn't blame you for. Anyhow, and then we're going to have our CH3 over here like this. Okay, now remember that the horizontal bonds are coming out, towards the page like this, and these ones are going in back. Now there's my Fisher projection, like that. But if we want to assign the stereochemical configuration at this chiral center, we're going to have to prioritize the four groups that are attached to the chiral carbon. The amine is going to have first priority because I have a nitrogen, carbon, carbon, and a hydrogen. Well, nitrogen has a higher atomic number than the other two atoms. Hydrogen is going to be lowest priority. I think it's pretty easy to see that this carbon is going to have second highest priority because it's attached to 3 oxygens. And this will be third highest priority. So if I follow this around like this, that looks like it's S because it's going counterclockwise. But remember, when your hydrogen is horizontal, you reverse it. Why? Because the hydrogen isn't going back into the page, it's coming out from the page like that. And so if it's going counterclockwise, this must have an R stereochemistry. So the stereochemistry of D-alanine is R, which tells us that L-alanine, naturally occurring alanine, must be S, right? Because it's opposite. It's not asking us for that, but anyhow. Let's try drawing the structure of, what's it asking us for? D-valine. Okay, let's give it a shot. So we'll start by drawing our amino group here. We have our carboxyl on the top. And then valine, I told you, has an isopropyl. Letter V is kind of the way you can think about it. So this would be D-valine. Highest priority, fourth highest priority, second, third, we follow. It goes counterclockwise. Our hydrogen is horizontal. And so D-valine also has a stereochemical configuration of R. And so if I had L-valine, it must be S. All right. Give me a thumbs up if you follow me on that one. Assigning some stereochemical configurations on a beautiful Friday. All right. Easy peasy. Good. It is easy peasy, right? Like, why couldn't more of organic chemistry been like this? Okay. Let's look at another problem. It says here, draw the form of the amino acid that is expected to predominate at the stated pH. Now, for this, you would have to have the table with all of the pKa values. I certainly don't expect you to memorize the pKa values of any of the protons in any amino acid. Okay, I'll write them down here. The pK of the carboxyl, so I'll put here the alpha carboxyl has a pK of 2.34, and the alpha amino group has a pK of, what is it, 9.69. I have them written down here in horrible handwriting. So if I'm at a pH of 10, that means that my amino acid is in an environment that's basic than most of these more basic than both of these pKa's. All right, so if I have an acid and it's got a pK of 2.34, well, if I'm in a more basic environment, then that's going to be completely deprotonated. And I'm in an environment that's more basic than the pK of my ammonium too, or my protonated amine, and therefore it's mostly going to be deprotonated. So I'm going to draw the bond line structure this time rather than the Fisher projection. So if I draw my alanine molecule, my carboxyl group is mostly going to be deprotonated, and my amine is mostly going to be deprotonated as well. Remember, the alanine just has a CH3, and so this is the form that's going to predominate at that pH. Let's try proline. Exact same strategy. I'm going to give you the pKa's of the alpha carboxyl. It is 1.99, and the alpha protonated amine has a pK of what? 10.60. That means that our pH is more basic than the carboxyl group, so that's going to be deprotonated, but it's more acidic than this, and so that is mostly going to remain protonated. Well, let's draw the bond line structure of our amino acid. Again, our carboxyl is mostly going to be deprotonated, but our amine will mostly be protonated like this. But remember that proline doesn't have a primary amine. It has a secondary amine, so we're going to be a little more careful here. And the R group is tied in a 5-membered ring like this. And so instead of having 3 hydrogens attached to my nitrogen, remember, nitrogen has a positive charge when it has 4 bonds and no lone pairs. And so this is the form of proline that's going to predominate at this pH. All right, let's try one more. It's glutamic acid. The alpha carboxyl has a pK of 2.19. The alpha protonated amine has a pK of 9.67. And the side chain, I'll just write it down here, side chain, because glutamic acid is a carboxylic acid is a side chain, is, what is it, 4.25. Okay, well, I think you're probably getting the hang of it by now. We see that this pH is more basic than the carboxyl. It's more acidic than the side chain, so that's going to remain protonated. And it's definitely more acidic than the protonated amine, so that will remain protonated as well. Let's draw the structure. We have our alpha carboxyl that's going to be deprotonated because a pH of 3, again, is more basic than 2.19. We have our amine, which is going to remain protonated, like that. And then we have our side chain, so glutamic acid has 1, 2 CH2 groups. And our carboxylic acid on the side chain is going to remain protonated. All right, give me a thumbs up if you feel like you could do that one. If I gave you the pKa's. All right, nifty. Well, let's pick up the pace a little bit. I think I'm probably going through these pretty slowly. It says use the data in the table with all the pKa's to calculate the isoelectric point of the following amino acids. Well, aspartic acid is a carboxylic acid, or sorry, has an acidic side chain, I should say. So it's alpha carboxyl. I'll write them a little quicker, is 1.88. Its alpha protonated amine is a pK of 9.60. And the side chain is a pK of 3.65. So our isoelectric point is going to be equal to the average of the two more acidic pKa's, or the two pKa's from the two carboxyl groups. So I'm going to take 1.88 plus 3.65. I'm going to take the average of that, so that's 5.53 divided by 2, 2.77, something like that. The next one is leucine. Our carboxyl group of leucine has a pK of 2.36. Our protonated amine has a pK of 9.60 for the isoelectric point. This isn't an acidic or basic amino acid, so we're just going to take the average, and we get 2.36 plus 9.60. We divide that by 2, and we get 5.98. Okay, and then lysine is a basic amino acid. Our alpha carboxyl is 2.18. Our alpha protonated amine has a pK of 8.95. And then the side chain is a pK of 10.53. And so its isoelectric point is going to be the average of these two pK values because it's basic. So we have 8.95 plus 10.53. I divide that in 2, and I end up with 19.48 divided by 2, which is 9.74. All right, there we have it. So calculating isoelectric point, not too bad. Well, let's get into the fun stuff, right? Let's talk about how you would go about making an amino acid. We look at a few ways. We look at some syntheses that are not chiral, and then we briefly look at a chiral synthesis. So you remember this reaction, the Helmholtz-Zielinski reaction, the HVZ reaction, was used to functionalize the alpha carbon of a carboxylic acid. That's this reaction right here. This was the HVZ reaction. For the first step was bromine and phosphorus tribromide, and then you treat that with water to hydrolyze the acid bromide that's formed. Remember, the book didn't even go over the mechanism of this. It just showed you some of the intermediates along the way. Well, what happens if you have an alkyl halide sort of here, if you treat that with excess ammonia, it's going to do an SN2 and displace that bromine to form an amino acid. Right now, obviously, since this is not a chiral center here, that means that you're going to end up with a racemic mixture, right? So you end up with a 50-50 mixture of the D and the L amino acid, or the R and the S, however you want to look at. One funny thing that the book brings up here, which I think is a great point for, if you're astute, you're really paying attention, you might say, hey, what about, you told me earlier on in the amines chapter, when we were looking at Hoffman elimination, that you could get exhaustive alkylation of ammonia. Remember that when you treated it with an alkyl halide? The reason exhaustive alkylation doesn't occur here, the reason why I only get one adding, is just because it's a secondary alkyl halide. It's just more hindered, so it's just going to react once. Would be a really good point to bring up, though. Anyhow, it's already brought up in the book. So that's one way. So basically, it says here amino acid synthesis via alpha haloacid. So this is number one. Let's number them. Okay, number one, amino acid synthesis via alpha haloacids. I mean, that's one way of describing it. I don't know. You could call it amino acid synthesis via a Hell-Volhard-Zielinski intermediate. I don't know. That's another way, because that way it would help me remember that it involves the Hell-Volhard-Zielinski reaction. I don't know. Anyhow, the next one is using the amido-malonate synthesis. You might remember the diethyl synthesis. We looked at that earlier on, the malonic ester synthesis using diethyl malonate, I should say. Anyhow, if you need to review that, it's in section 21.5. We looked over that in dirty detail. But if you modify the diethyl malonate, so diethyl malonate was just what I have in the red circle. If you put this, N-acyl group on here, okay, then you get this new molecule called diethyl acetoamidomalonate, or diethyl acetoamidomalonate, however you want to pronounce it. Anyhow, it's kind of a clever way to synthesize an amino acid, right? Because you have an acidic hydrogen here. You can treat that with sodium ethoxide, right? Because it's got a low enough pK. You get complete deprotonation. Then you get alkylation. Do you remember what happened here? Right, when you heated this, you converted both of these esters to hydroxyl groups. Well, if you treat it with acid and you heat it up, not only are you going to convert both of those to carboxyl groups, and then you get a decarboxylation to form the carboxylic acid, but it's also going to hydrolyze this acetyl group off to give you the ammonium, okay, to give you the protonated amine. So it's kind of a triple whammy, right? It does the hydrolysis of these two esters, converts them to the carboxyls. You get a decarboxylation. You make the carboxylic acid, and you hydrolyze this all at the same time. It's really a thoughtful synthesis, in my opinion. Okay, very, very thoughtful. Whoever came up with this is pretty cool. Okay, but again, you end up with a racemic mixture. Let's say you only wanted to make, some kind of new L-amino acid. Well, now you got a 50-50 mixture. How are you going to separate it? You know from the lab that separating compounds isn't that easy. And separating amino acids, I'm not even sure. It doesn't sound fun. Okay, I know that in the book it says column chromatography or something, but you'd have to use chiral column chromatography to separate those, or maybe a supercritical fluid or something. Sounds miserable to me. Anyhow, what if you wanted to make phenylalanine, okay, using that amido-malonate synthesis? What would you do? And I'm definitely going to ask this on your next quiz. How would you use the amido-malonate synthesis? What would your choice of alkyl halide be? Well, this portion of the molecule, all of this part here that I'm boxing in red, right? That comes from this starting material, okay? This portion, the R group, this comes from an alkyl halide. Alkyl halide. So what you've got to be able to do is, depending on the R group, you've got to be able to choose the correct alkyl halide. So in this case, it would be benzyl bromide, okay? That's going to react in the SN2 reaction, and then you hydrolyze everything into your decarboxylation to make a racemic amino acid. Pretty cool, huh? And we're going to look at questions that deal with this. How would you do this? What would the alkyl halide be? Another way to synthesize a racemic amino acid, and this is an old reaction too, is to use the Strecker synthesis. It's a two-step synthesis from an aldehyde. Aldehydes are pretty abundant. If you can't find the aldehyde, get the alcohol and just oxidize it using manganese oxide, right? Or PCC. All right, so you oxidize that to an aldehyde. Then you can treat that with ammonium chloride and sodium cyanide and make what's called an alpha amino nitrile. And hey, everybody in the class knows this reaction, right? we looked at the hydrolysis of a nitrile to a carboxyl. That's something we looked at in the carboxylic acids chapter. So really the only new thing here is going to be this step, taking the aldehyde, treating it with ammonium chloride and sodium cyanide to make the alpha amino nitrile. And the mechanism for this is shown in the book. You don't have to know the mechanism. I'm not going to ask you the mechanism of the alpha amino nitrile formation. But what I will ask you to do is to use the Strecker synthesis in order to synthesize an amino acid. And so you need to be able to choose the correct aldehyde. For example, what if you wanted to make alanine using the Strecker synthesis? In that case, you would use ethanal or acetaldehyde. All right, you treat that with ammonium chloride and sodium cyanide, make the alpha amino nitrile, and hydrolyze the nitrile to the carboxylic acid. There you go. But again, it's a racemic synthesis. You end up with a 50-50 mixture. The mechanism for the formation of the alpha amino nitrile is shown here. I'm not going to go over it in detail because I've never asked it before and I'm not going to ask it now. It's not going to be asked on an ACS final. It's not going to be asked on MCAT, but it is an important reaction. What if you wanted to do a chiral synthesis, or what we call an enantioselective synthesis of an amino acid? That means, what if you only wanted to end up with one product? You don't want to get a racemic mixture. Well, again, everything that we've looked at so far provides a racemic mixture. There's a couple of ways. Okay, you can either do a resolution. That means you separate the enantiomers, which, depending on the situation, and I'm not an expert, I'm an expert in separation. I've done enough of those. But chiral separation or separation of racemic mixtures, I worked in a program at the University of Montreal where we did a lot of separations of enantiomers, but those were not done, generally speaking, by the chemists in the lab. They actually had a separations expert. And I don't know if you remember phase diagrams from general chemistry. You remember the phase diagram, the solid, liquid, and gas. And then once you get past the critical point, it becomes a supercritical fluid, which is like not a liquid and not a gas. Okay. Well, what they can do is do a type of chromatography using carbon dioxide as a supercritical fluid, and that can separate enantiomers. So it takes special training in order to operate the machinery to do that. But the best way would be to not even end up with a racemic mixture to begin with, right? Would be to do an asymmetric synthesis. So that means make only one enantiomer. Sometimes I call that a chiral synthesis, right? You're making a chiral compound. It is clearly more efficient to synthesize the desired enantiomer as opposed to fishing it out of a racemic mixture. Now, it depends on the situation. Maybe you don't care if you have a racemic mixture. Okay, no problem. But generally speaking, chiral synthesis is going to be much more useful in organic chemistry. So how would you do that? And we don't, I'll be honest, in this class, we don't look at a whole lot of chiral or enantioselective synthesis. But the way you would do it is you would use what's called asymmetric hydrogenation. Now, it's just like our regular hydrogenation, but the catalyst that you're used to with hydrogenation were what? Palladium, platinum, or nickel to hydrogenate a double bond or a carbonyl, for example. But if we want to do an asymmetric hydrogenation, we use hydrogen and what's called a chiral catalyst. Now, if you look in our book at the structure of a chiral catalyst, you'll see it's a pretty interesting molecule, and I have a picture of it on the next slide. But what you're going to do is you're going to asymmetrically hydrogenate this pi bond, okay? So that means you're only going to put the hydrogen in from the back face of the molecule. So the hydrogen is going to be in the back, right? And then your nitrogen is going to be in front. You're going to blow this acetyl group off here and these ones too. And this would be a synthesis of the molecule L-DOPA. Anyhow, so many chiral catalysts consist of ruthenium. Now, I know ruthenium isn't an element we think of a whole lot in chemistry, but ruthenium complex to what's called a chiral ligand. And so this is the chiral catalyst here. I'm not going to, it's one of those things I could ramble on about for quite a while because, you know, you might look at this molecule and say, well, I don't see any chiral centers in there. How the hell is that chiral, right? Why is it rotating plane polarized light in a specific direction? Well, there's a good reason for that. The reason why it's chiral is because its rotation around this bond is restricted. And so it is, in fact, a chiral molecule. If you draw the mirror image, And since there is no rotation around this bond, it is in fact chiral. So it's a different type of chirality than you're used to seeing, but it is chiral. So if you wanted to make, for example, diphenylalanine with 99% enantiomeric excess, you have a pi bond or a double bond at the alpha carbon, and you protect the amine as an acetyl. So AC, and you probably know this by now, is just an acetyl group like that. Okay, you're going to treat it with H2 and the chiral catalyst. So chiral catalyst, and you're going to get a really high enantiomeric, or make the product in a high enantiomeric excess. Then you just hydrolyze off the acetyl group, and you end up with diphenylalanine. Again, for our class, I'm not going to ask you to draw this out, but you should be able to recognize that when you see that it is a chiral catalyst. And if you want to, read up on this, you're like, I have a thirst for knowledge. I need to know more about chiral catalysts. Definitely, our book does have more information about it. We didn't cover it in Organic Chemistry 1, but you should know just a chiral catalyst, and there you have it. All right, well, that's a lot of information covered. Let's switch gears and try some practice problems. I got a bunch here. I really have a real pile of them before we get into peptide synthesis in the second part. So I don't know if I have time to go through all these practice problems, but we'll look at a few here. It says, identify the necessary reagents in order to make the following amino acids using a Hell-Volhard-Zielinski reaction. Well, remember that this is going to be a achiral synthesis, right? We're going to get a racemic mixture. Now, first, if we want to look at the structure of leucine. So leucine, you remember, is when we had an isobutyl group as our R group. So I'll just draw the structure of leucine here. I'm going to leave the chirality out of it because this is going to be an achiral synthesis. And then we're going to have, right, we're going to have an isobutyl group. So this portion here is an isobutyl group. Well, if the Hell-Volhard-Zielinski reaction involves putting a bromine at this position of a carboxylic acid, that means the carboxylic acid we're going to want to start out with is going to look like this. We're going to have our carboxyl. We're going to have 1, 2, 3, 4, and something like this. Right? So all I did was take this part of the molecule right here because I'm going to put my bromine where the amino group is going to end up, won't I? Well, the first step is going to be to treat the molecule with bromine and phosphorus tribromide, PBr3, as my office mate used to call it, and then treatment with water. That's going to give us the alpha bromo carboxylic acid, isn't it? So now we've got something that looks like this. And now we can do the alkylation reaction by treatment with excess ammonia. And we're going to end up with this. Now, if you wondered, what the heck is that? What the heck is that squiggle? The squiggle means that it's a 50-50 mixture of up and down. Okay? Another way of saying that would just be racemic. So we end up with a racemic mixture. Okay? And you can try the same thing with alanine. Alanine's an even simpler amino acid. The R group in alanine is just a methyl group. So if we want to make this compound, we're going to start with propionic acid. We're going to treat it with bromine and phosphorus tribromide. Hydrolyze that. And then the third step, we'll treat it with excess ammonia. And that's going to give us racemic alanine. Not a chiral synthesis. Let's try another one. Each of the following carboxylic acids was treated with bromine and PBr3, followed by water. The resulting halo acid was then treated with excess ammonia. Draw and name the amino acid that's produced. Again, I don't expect you to memorize the names of all the amino acids, but all we need to know is that based off of the last question, what are we going to do? We're going to put an amino group here, and we're going to put an amino group here. So after all of that spinach, bromine, phosphorus tribromide, you can see why this is the dessert chapter, right? Water and excess ammonia. All you have to know is this. you're going to end up with a carboxylic acid, or sorry, an amino acid that looks like this. We're going to have our amine here, and we have like that. This is a leucine, and if we do the same reaction conditions here, what amino acid are we going to get? We're going to end up with valine, aren't we? So when we have an isopropyl, that's a valine. And we must point out that both are racemic. It says here, if we wanted to use the amidomalonate synthesis, for which we have this starting material, so we take the diethyl malonate, we add a nitrogen with an acetyl group on it like this, so this is going to be our starting material. Okay, how would we make the following amino acids starting with this compound? Well, first, let's look at the structure of isoleucine. Isoleucine, you probably remember, is an amino acid that has 2 chiral carbons. We've got our carboxyl, our amine, and isoleucine has a sec-butyl group coming off it. So there's a chiral carbon here too. Now, it doesn't say anything about L amino acids or D amino acids, so we're going to end up with a mixture here because this is not a chiral synthesis. But remember that from this molecule here, okay, our starting material, what parts of the amino acid are going to come from that? The parts of the amino acid that will come from that are going to be all of this part of the skeleton right here. So that tells me that this part is going to have to come from my alkyl halide. All right, well, let's take a look at what that would look like. So we'll start with our starting material. That's redundant, isn't it? Copy. So we have our starting material. We're going to treat that with sodium methoxide, followed by the appropriate alkyl halide, 2-bromobutane, or 2-iodobutane, doesn't matter. What's that going to give you? It's going to give you this molecule with this attached to it, isn't it? So then, when you treat that with acid and you cook it up, you again hydrolyze both of the esters to carboxyls, you do a decarboxylation, and you remove the acetyl. And since you're in acid, you're going to end up with the protonated form of the amino acid. So you're going to end up with isoleucine in this form, where the amine will be protonated. And there you go. Isoleucine, I'll use the abbreviation ILE. For alanine, alanine has only a methyl group, and so the alkyl halide that you would use for alanine synthesis would be simply methyl bromide or methyl iodide or methyl chloride. And then for valine, it's got an isopropyl group, so the alkyl halide would be isopropyl iodide, isopropyl bromide, doesn't matter. We'll choose the bromide. The same type of synthesis, the amidomalonate, was performed using each of the following alkyl halides. In each case, draw and name the amino acid that was formed. This is in the exact same vein as the last problem. We have our starting material, and we treat that with sodium methoxide, followed by methyl chloride, CH3Cl. What are we going to end up with? This compound with a methyl group attached to it. So when we hydrolyze that with acid and heat, we're going to end up with this amino acid. Our carboxyl group, our amine is going to be protonated, and we're going to have just a methyl group attached. Could anybody name that amino acid? I'd say it's the second simplest amino acid. Yeah, it's alanine. All right. If you try the same thing with isopropyl chloride, which is this molecule, then your R group is going to be an isopropyl group, which means you're going to have valine. And if you were to use 2-methyl 1-chloropropane, you're going to end up with an isobutyl group. How should I draw it? Like this, right? An isobutyl group, which means you're going to synthesize leucine. All right. Well, we're running out of time. I expect you to look over and do all of the problems in the chapter before coming to class on Monday. We'll finish up this problem, and this problem. And then we're going to tackle the second half of the chapter, which deals with the structure of peptides, sequencing a peptide, peptide synthesis, and then we will very briefly look at protein structure.